Let's get oral.
Hello. Welcome back to Let's Get Oral. I am Camille with my co-host Jonathan. Hello, Jonathan.
Howdy.
Hi. And today I'm really excited to introduce Dr. Keith Kirkwood, who is a longtime friend, coworker, although I don't think I ever worked with you underneath you, but definitely from across the nation when you were down in Charleston. You came from University of Buffalo, then went to West Virginia, and then came back?
A little bit different. Hi, everyone. So I'm from Buffalo. I actually am from West Virginia. I shuffled off to Buffalo. I was tenured at Michigan, and then I went down to Charleston for almost 10 years. And it's actually been about 8 or 9 years that I've been back in Buffalo, where it all started. So I'm happy to be back.
Yeah, the weather's not killing you?
Not so far.
Oh,
so
good.
It's good. No, you get used to it. Buffalonians are quite used to the winters here. So they just last a little bit longer than everywhere else in the country.
Yeah, that lake effect is real sometimes, isn't it?
It is.
Well, I'm happy to have you on. I've followed your work for a long, long time. And so to give an awareness to the crowd, this is a dentist, gone periodontist, and then gone full osteoimmunology, so bone health, but with a flair of the immunological aspects and how it relates to oral health. All very exciting stuff, especially right now when we're starting to really take oral care as a part of our longevity or metabolic health. And so your recent paper really caught my interest. And before we get to that, I just want to ask you how you fell into this very niche space. Give us a little taste of your journey.
Well, I've had a long journey. And when you ask me this, it feels like I have to reach back to a previous life at this juncture in my life.
I bet.
So I've had a long time interest in alveolar bone loss, associated with periodontal disease, because it is a hallmark of periodontitis. And so how this happens, and then therapeutically, how can we attenuate that, mitigate bone loss associated with periodontal disease has been sort of, my niche for a long time. So we've become interested in obesity-associated bone loss with periodontitis. This paper that we're going to talk about more is really about long bone, but it has implications, and we've previously published things around oral health and this particular protein that we're going to talk about several years ago, in fact. So I've become interested in really the intersection of immunology and oral bone loss from probably the beginning of my career. So.
Yeah, I remember coming in quite a few years ago. Maybe you were mid-career at that point. But I think that we were all very focused on the oral bone loss, the maxillofacial bone loss. But there is an element of total bone loss that comes in aging. And is that related or increased with periodontal disease? Do we have kind of a delineation between the two events, or is it all taking place at once?
So the periodontists that are listening are going to be telling you that, aging by itself is not a risk factor for periodontal disease. But as our immune system ages, right, then it's clearly associated with alveolar bone loss, especially in mice. And so we can see these changes that occur irrespective of periodontal status. We see bone loss. And we see it, the same as in long bone, appendicular bone. We see it with oral bone and alveolar bone loss as well. So they're intertwined, and they're intertwined because of the osteoimmunology that you mentioned at the beginning. The osteoclast is the only cell in your body that can resorb this bone. It's housed, or it's derived, all immune cells are derived from the bone marrow. So, how this is happening, how it's intertwined with oral bone loss is really the intersection that is this osteoimmunology, and that's where I spent most of my career. So it's definitely part of the mix. Aging by itself is a risk factor for periodontitis. People who are older have more disease burden. That is very, very clear. And it's not simply a chronological age. It's immunological age as well that impacts this.
How do we define immunological age?
That's a tough nut to crack. There are several papers out there that really speak to, when we have chronological age and then, accelerated changes in immunological aging or biological aging that come in spurts. And so that evidence is there. I'm sort of at that place in my life chronologically where I have these biological aging impactfulness, and I'm sure we're all going to go through this, but they happen at certain periods in our lives. So whether or not that biological age is tied in with oral bone loss remains to be determined. But we certainly know that with age in general, we see these changes. So there's an intersection there that we fully don't understand as yet.
Right. So, this is something that we talk a lot about in facial aging in general, is that you get global bone loss. And so how that pans out is all of the holes in your skull get a little bit bigger. Your ocular socket, your nasal, all of those widen. And that's why you get kind of a cavitation of your eyes, and they kind of recede back into your skull a little bit, which is then why we just have a overall different appearance. But then there's this other phenomenon where we have a widening of the jaw. Even though there is bone loss there, it is taking a slightly different shape. And I think it feels more exaggerated in women due to the fact that we typically have these more feminine features that then tend to look more masculine as we age. But can you speak to some of those differentiating factors?
Probably not, Camille. I think that when we model stuff in preclinical models, it's very, very different in humans. And I think even in the paper that we're going to talk about, we're modeling sort of frailty and long bone, and that's a different kind of bone, literally, than the predominantly intramembranous bone or facial bone, oral facial bone. So I don't think I can really directly speak to how this paper would intersect with that. And I really wouldn't be able to be well.
No, that was a wild card question.
That.
I like to postulate that it has more to do with mastication and use of bone.
Yeah, I think that's what I was thinking about when you asked this. I'm sure it has to do with, vectors of force over time that, lead to these changes.
And then you get the recession around it. And I think that.
Well, I think it does speak to part of what we're talking about today, which is bone is a very dynamic tissue. It's not static.
Right.
And it turns over roughly 3 times every year. And so we have a tissue that manifests, but it's certainly not the same as where it was 20, 40 years ago. And so I think all of these things impact aging, obesity, this protein we're going to talk about. But I think at the end of the day, it just speaks to the fact that it's a dynamic tissue.
So I asked you about the immunological age, and I think that your protein here kind of plays right into that, honestly. So I read it as tristetrapolin. Is that right?
No, tristetrapolin.
Yes, there we go. Or I'm going to, for the rest of this,
TTP.
say TTP. Yeah. So
thank you.
Tell us about this protein. What is it?
Yeah, so TTP is a RNA binding protein. The gene encoding it is a zinc finger binding protein, ZFP36 in this case. It has a family of other zinc finger binding proteins, which are RNA binding proteins. And the way it works really is it's sort of the brakes on inflammation.
So, what's TTP? TTP acts like the cleanup crew for inflammation. When our immune cells make messages telling the body to produce inflammatory chemicals, TTP helps destroy those messages once they are no longer needed. So when we don't have enough TTP, these inflammatory signals can linger around, which then further encourage our immune cells to break down the bone in that local area, which can then further become overreactive. So when this is happening around the teeth, the lack of TTP could contribute to the bone loss that's seen in periodontitis. Now, back to Dr. Kirkwood.
And so it regulates RNA, not transcription, but translation at the level of mRNA stability. So it really promotes rapid degradation of transcripts, which are primarily pro-inflammatory.
Right. So what you're saying in the layman's way, I'll reiterate, to say that it's a protein that binds to the RNA that is going to give you an inflammatory protein. So it's binding that up and preventing that inflammatory protein from being made. Yeah?
Right. So I would look at it this way. So we don't want a lot of inflammation around.
Correct.
And so that would be deleterious to the host, including the oral cavity. So if the decision is made within a given cell to make these particular transcripts that are pro-inflammatory, we have to be able to control that ability to even make that protein. So it gets back to the sort of central dogma of life, which is that DNA is transcribed into RNA, which is translated into protein. So TTP is more focused on RNA decay, and it basically targets these segments of the messenger RNA within the cytoplasm that sort of tags it for rapid decay. And so once TTP binds, it then triages that mRNA to be degraded within the cytoplasm. So think about it as sort of like a rheostat.
Right.
So you have the accelerator with transcription, all right, and within the nucleus, that mRNA goes into the cytoplasm, and then it's got a fate to be decided by TTP and other RNA binding proteins that act in a similar manner. So these targets are really there to help prevent this sort of blazing fire. So these are like little bursts of like a flare, if you will. And so TTP controls the numbers of flares so the fire doesn't go out of control.
What regulates TTP, though?
Phosphorylation. So phosphorylation is controlling the ability for TTP to bind to this particular transcript, to even be located within a given spot within the cytoplasm itself. And so this is all sort of, it gets back to periodontal biology, where largely gram-negative bacteria and the lipopolysaccharide, or LPS, trigger an immune response, and the cell signaling that happens is going through primarily, in my case, P38 MAP kinase, which then can phosphorylate directly or indirectly through its downstream target, MAPPAT kinase 2 or MK2, and the biology is pretty well understood. But TTP is a protein that targets these transcripts and controls inflammation at the level of mRNA so that basically can be reversed quicker. So you have these breaks that are sort of, there. What happens with aging and TTP is basically the breaks wear out. And so you have the situation where TTP expression goes down with age. And so the breaks have been sort of lost. And that's why you have an increase in what we're really calling this infla-aging, which is sort of increase in inflammation that is associated with aging, and sort of sterile inflammation because it's not induced by a virus or a bacterium. So this sterile type of inflammation goes in tandem with sort of the aging, the biological aging of the immune system, which is often called immunosenescence. And so that sort of runs in parallel with this infla-aging. So once you have this infla-aging with all the pro-inflammatory cytokines that are increased, you concomitantly or at the same time have a decrease in sort of the plasticity of the immune cell populations, especially T cells. So T cells, are decreased in terms of how they recognize things, and their repertoire decreases. At the same time, you have these sort of decrease in the lymphocytic population. You have an increase in these myeloid cell populations. And the myeloid cells sort of overtake that space within the bone marrow. And so this population that we've been interested in, which are myeloid-derived suppressor cells, sort of sit in this middle of all of this aging-related sort of hallmarks of immunosenescence. So MDSCs go up, these myeloid-derived suppressor cells. The name comes from the cancer biology arena, where they suppress tumor immunology and suppress T cell repertoire within that setting. In our setting and context of periodontitis, they are a precursor population to make an osteoclast. So there's a lot of different ways that you can become an osteoclast, a lot of different precursor populations. MDSCs, we're interested in this population because they're increased with aging and they're also increased with obesity. And so we're very interested in sort of understanding the mechanisms, which are different, of how this kind of intertwines with aging in the periodontium and long bone, and how this predisposes people to more bone loss, whether that would be associated with long bone or associated with oral bone. So it's very much this sort of pivotal breaks on the system that's lost.
So in a previous paper, you showed that the lack of TTP led to exacerbated bone loss. Can you walk us through that and talk about any observations in the oral cavity at that time?
Yeah, that was completely by luck. So let me go back to kind of explain to the audience what this animal looks like with the loss of it. So we use genetically altered models. So the knockout for this, where the gene is knocked out sort of globally within the animal, is lost. And so what happens in that situation is there's a lot of inflammation, mostly driven by TNF alpha, but we've looked at interleukin 6 and IL-1 beta, and they're also elevated. And all of this work really happens not from all of my lab, but from the seminal discoveries of Perry Blackshear, who's a co-author on this paper we're talking about today. But Perry's lab was the first to both clone that gene as well as make the knockout animal, and he's at the Research Triangle at the National Institutes of Environmental Health and Science in the Research Triangle at Duke. So his observations, kind of led us to understand more about oral bone loss. So we had the knockout, back to the story. We had the knockout animal, and we usually used, at that time, direct injections intraorally of LPS to initiate bone loss.
I'm assuming E. coli LPS.
No, we used Actinobacillus at the time,
Okay.
Agrobacter. So we used oral bacterium to cause bone loss. And what happened, because these animals were so pro-inflammatory, they had cachexia, which is almost like being septic. So we would hit them with one injection intraorally with LPS, and all of the animals would die. So we didn't know what to do. And
it's an over response, just clarify.
It was an over response. So basically they got septic and died very quickly within 24 hours. So honestly, it was serendipity. We just sort of let these animals age in the vivarium. And over about 9 months, we're like, well, what do we do with these guys? And so let's look at the bone loss. And so we looked at long bone and we looked at oral bone, and both, without any perturbation, without any, experimental periodontitis model, there was less bone. And so I had a postdoc at the time, Heidi Steinkamp, who made this observation, and she then did the immunophenotyping on the animal and showed that the MDSCs, this monocytic population that becomes an osteoclast, is increased. At the same time, we had a decrease in T cell populations, both CD8 and CD4 T cells. So it had an immunosuppressive phenotype with an increase in myeloid cell that becomes an osteoclast. There was more osteoclasts. So we began to think about, we published that paper several years ago. We also did the reverse. We overexpressed TTP and blocked LPS-induced bone loss. And so that became sort of this loss and gain of function. So with the present paper, we really asked a different question, which was, if we prevent aging-associated loss with this TTP using an animal that's a knock-in approach. So TTP, interestingly, autoregulates itself. It has this region within the 3' untranslated region where there's these AU repeats. So these are AU-rich elements where TTP can bind. So it autoregulates itself. So Blackshear's lab made this knock-in animal where it overexpresses TTP, but not by the traditional sort of very much overexpressed situation using these promoters that drive expression very, very heavily. So this was more of a natural response.
Almost like a turning off the brakes a bit.
Correct. So we don't have the ability for TTP to be sort of turned off like it usually is with the aging process. So what we have is TTP being sort of regulated by its endogenous gene promoter. So it's in sort of a natural setting, if you will, and it's not being overly expressed like we did with the perio model several years before this. So in this case, we're not really looking at it as a therapeutic, Camille. What we did was we looked at it, I would say, more from a preventive model, because we aged this animal. And so what we found was that at 6 months of age, between the knock-in and a wild-type animal, we really didn't have any difference. And we collaborated with another lab to really look at frailty of bone. So this is not really an oral health question per se. It's more of a aging gerontology question. So we collaborated with Bruce Trone, who at the time was here at the University of Buffalo and since has moved to Kansas City, KUMC, the medical center there, in charge of an aging center. And so Bruce's lab did a lot of these frailty assessments. So my lab didn't do that. So this is an example of collaborating with people outside oral research domain so that we could understand how, in this case, how TTP overexpression with this knock-in animal prevented aging-associated frailty of long bone. So we used a bunch of different composite scores. They put them on a treadmill. They used grip strength. They basically did not really cognitive function, but physical activity. And so what we observed in both male and female animals is that we had protection against aging-related demise, which was incredible. And then on top of that, it sort of reversed the immunological phenotype. So we had the intersection of frailty assessments, which were lessened, as well as immunological changes. With the male mice, we saw changes in the bone, but in the female mice, not as much. And probably a lot of that has to do with the already decrease in ovarian function or loss of estrogen at the time these animals were used for the experiment. So we compared sort of adult animals, 6 months of age, which is sort of like in your 30s or 40s in a human, compared to 22 months of age, which is about 65 to 70 years of age in a human. So we compared sort of an adult with an older adult. I don't want to say old because I'm rapidly approaching that time frame, so I don't want to say that because it seems a little too personal, I guess.
It seems less old now that you're closer. Because we still always feel fine inside.
Indeed. We're always a child inside, exactly. So I think, that whole story of how TTP sits in this center of, infla-aging becomes really important. So if we reversed it, we saw parameters that were enhanced that were more like a more youthful phenotype or appearance. And the immunological changes were astounding. And I think, that's sort of the nuts and bolts of that paper.
That is exciting. And that's what makes me want to go back to and really understand the regulation of TTP inherently. So you mentioned phosphorylation. So maybe the bacteria is stimulating our immune cells and we're getting an upregulation of phosphorylation through those pathways. And that initiates TTP to control the fire, if you will.
Correct. So the biology of this is pretty cool, I think. I mean, because I've been doing it for so long. So imagine a transcript that's very short-lived and you don't want it around like TNF alpha or IL-1 beta, these pro-inflammatory cytokines. So TTP is basically sitting on that 3' untranslated region and promoting rapid degradation through a couple different mechanisms that, no one here is going to care about. But it's basically promoting degradation to keep that pro-inflammatory signal at bay, putting out the fire, so to speak. When you have a inflammatory insult, like in the oral cavity where we have periodontal bacteria that have LPS and stimulate cell signaling, that's when that phosphorylation signal is there. So when it's phosphorylated, and we know the residues that have to be phosphorylated, that TTP that's sitting on that transcript sort of comes off of that transcript. And that allows for translation of that protein and increased expression. So the brakes are basically coming off. And so that transcript is being made more heavily. And so that does happen in humans and in mice in periodontal situations. And so, we've known this for over 15 years, I guess, now. So this paper, again, is just sort of reversing this to ask, I guess, more of a therapeutic question about if we change this transcript ability to autoregulate itself associated with age, will we reverse this? So that was really the premise of the question.
So we're not going to be seeking a therapeutic in this space. So how could we?
Yeah. I think, therapeutically, right? We don't know the answer to that. Blackshear had done what's called chemical library screens to look at things that would increase TTP expression. We tried those in the lab. Really, none of those really worked in our hands. So the other thing to do would be because what we did with these animals with a knock-in, that's really not translatable into humans at this point. So, you can think about it as increasing expression of this protein, but, if that did it sort of globally, I don't know what would happen. I think if you did it in the particular cells that we're interested in, more in what we call the myeloid cell, lineage cell, that might have traction in terms of that. And I think that, probably the way that we would do it would be to maybe control the phosphorylation, maybe control that. And that's it's kind of coming full circle in my career now, because we essentially looked at signaling pathways after LPS stimulation many, many years ago. And we argued that if we target these, let's say, positively activating pro-inflammatory signals, such as this MAP kinase P38, why wouldn't that have utility as a therapeutic to attenuate periodontal bone loss? And in mice and rats, it did. Unfortunately, most of those small molecule inhibitors are very, very toxic. They cross blood-brain barriers. They have a lot of skin things. And there's still sort of these pipeline drugs that are out there, but have not come to fruition in terms of FDA clearance, right? And these would be more geared towards, the market's too small for periodontal disease, unfortunately. We'd all like to think it affects 47% of the US population, but that's apparently not good enough for our market. But I was being facetious. But the idea is that it would be potentially used for things like rheumatoid arthritis. And
right,
so that would be, where people would get more excited, apparently, than controlling periodontal disease. So I'm biased, as you can tell. So I think the idea is that, we would control phosphorylation because these other drugs that would decrease the signaling mediators associated with that phosphorylation, they've all been toxic. And so they're just sort of a no-go. But the idea is there that they too, just like targeting TTP, they would block a number of pro-inflammatory cytokines as opposed to just sort of targeting IL-1 beta or TNF alpha or IL-6, which we do have immunotherapies against those already. We would hit it sort of further upstream with the signals or the targets of those signals that would regulate these pro-inflammatory molecules that would have more potential therapeutic benefit than just targeting an individual one. That was sort of the goal very, very early. We've tried various ways of doing it in preclinical models. I think the idea is to make that translation happen. Local drug delivery could still be an opportunity. We've actually, utilized things where we've encapsulated MAP kinase inhibitors into nanoparticles and delivered that. That's been efficacious in preclinical models. But none of these really targeted directly anyway, TTP. It would be sort of indirect targets. I think the idea with a lot of this and a lot of what I've done that, has been graciously funded by the NIH to date has really been, sort of proof of concepts, right? Proof of concepts mechanistically that these targets are important for either homeostasis if it's not around, or if you overexpress it, potentially a therapy that could be used to reverse bone loss. With this paper in particular, back to the paper, I guess, with the younger animals, the 6-month-old, we saw no difference. So this was kind of cool because it doesn't mean that you overexpress this, everything's going to be better. It's not going to be like this sort of fountain of youth, right? Even at middle age. It's going to be mainly targeting the effects of loss of TTP that happen with age. So it is more of an aging specific opportunity. But right now, if you ask me what the next steps would be translationally, it would probably fall a little bit short at this time because we don't have targets. We understand the mechanisms, but we don't have the therapeutic opportunities to target these specific types of phosphorylation events at this time.
I want to kind of come back to the prevention idea and say that I see the opportunity in prevention, and then the oral cavity becomes like the linchpin, if you will, because the oral cavity, is your first in line to your metabolic health. And we know that as we age, that would be the first signal that we get before it then rejiggers our intestinal flora and then proceeds to create leaky gut and metabolic disorders. Some of Glogauer's work has been really interesting to watch. He's basically finding in his multitude of studies that the myeloid cells change or altered under periodontal disease. Like once you click over to that periodontal disease state, then your immune phenotype will change. And it seems to be a permanent alteration. And so if the prevention would be to maintain a level of health there, prevent so much phosphorylation cascade, and keep that TTP in check, if you will. Mm-hmm. And is that how you would go about it?
No, I think you hit the nail on the head, Camille, because that would be, I think once the deleterious effects happen already with immune changes, the memory either, sort of genetically or epigenetically modified, that would be sort of hard to reverse. So if you think about it as a therapeutic, it would have to be almost as a preventive because you're trying to prevent these things from occurring. I think the challenge there is not understanding the mechanisms, it's understanding what kinds of therapeutics can be used in that scenario, which would be sort of like anti-aging, if you will. Right. You did mention one of the more important aspects is that, with the loss of this protein, we have this inflammation, or sometimes it's called sterile inflammation, right? And because it's not triggered by bacteria or viruses. But the microbiome controls a lot of this increase of these myeloid-derived MDSCs. So we know this from other studies that we are publishing, submitted papers or to be published relative to periodontitis, that this controls a lot of it. So, it does emanate or start to come from the changes in the gut. And so if you think about the changes associated with obesity, and that's what a lot of our focus has been, but even with this sterile inflammation model of loss of TTP, we've recently done some sort of animal studies where we co-house these animals. And because the animals are corporophagic, which really means that they eat each other's fecal content, trying to keep it clean for the podcast.
You could say poop.
You can say poop. Okay. So they eat each other's poop. And so that microbiome is distributed. So if we co-house wild-type animal with knockout or knockout with wild-type, we see changes in the microbiome. We also see changes in the immune system. and we see changes in the bone phenotype. So we've kind of come full circle with, and we do have germ-free animals as sort of controls for all of this. So we've really kind of looked at the changes.
So wait. I'm going to stop you there. I was really hoping you would bring microbiome into this because that was going to be my next question.
I know. Yeah, I know.
So with the...
I know your background.
With the knockout and co-housing, do you know the microbial story in any detail there?
It's to be determined. The paper's been submitted. We'll send a revision next month. We do, Camille, but it's sort of the mouse microbiome, which it's not exactly human, and it varies between animal facility. So that would be more of a change from, let's call it healthy plane, to something that's more disease-like, but it does drive the expression of these myeloid lineage cells. So
the end result would be the wild type.
Yeah, it's kind of like chicken or egg question, right? You have to understand the inflammation drives the changes in the gut, but the inflammation in the gut's what really drives everything else downstream of that. And so that change with obesity, with aging, with this loss of TTP. So the loss of TTP became an opportunity to understand this from a sterile inflammation perspective, not anything else.
Yeah, and I think that is so important because I feel like this is understated in the dental community, specifically around the microbiome, because we just think, oh, well, if Porphyromonas gingivalis is there, or, the treponema, whatever, but how it flourished in the first place.
Right. No, I think this is the first part of the digestive system, and there is communication between the oral cavity and the gut, and vice versa, right? It goes both ways. We've done study with Crohn's disease. Patricia Diaz, who is here, you should have her on. She's done some stuff with type 2 diabetes and transmission.
She's great.
So we're basically understanding more that whatever perturbation we have on a system that affects the oral cavity will impact the gut, and that also has reciprocal implications as well.
Right.
And I had a former postdoc in my lab who looked at germ-free mice and sort of wild-type animals just in a specific pathogen-free environment that we house animals in, and we see differences in alveolar bone. And so we really haven't necessarily properly even defined homeostasis, right? So we can't get away from the microbiome, although sometimes I would really, really like to, unlike you. But the microbiome helps define everything in the immune system, and it is seemingly the driver. So what I tell students and what I tell people in my lab is that we live in their world, and they've been around a lot longer than we have. And so they dictate a lot of what's happening within our bodies in terms of what the immune responses are, and how ready we are to be poised to respond to these immune responses.
I don't necessarily think they're drivers. I think, to your point, you co-housed in a non-infectious model, and you saw bone loss in the wild type that you weren't seeing in your regular wild type. And I think that this is a really important point. That there's a microbial response to having a change in your genetic code.
Right. So you have hyperinflammation, and that drives a change. So,
yes, and then that change can pass on.
Right, but the microbiome change is what really drives the change in the immune system. So it is a vicious circle. So
it
it's hard.
It absolutely is.
Yeah.
And I want to clarify that these mice are eating poop. It's not kissing. They're not kissing. They're eating poop. So I would like to make that distinguishing feature right now.
And I hope that doesn't translate into humans. But I'll leave it at that.
So for prevention, that's off the table.
Right. Literally.
So what are you doing for prevention?
What am I doing for prevention?
Mm-hmm.
Well, first of all, I'm not corpophagic, so I think that's okay.
We're all good.
We're all good that way. I think, it's a balance of it, like everything else. We can't control everything, and I think that's important that we all understand that. I think just the hallmarks of oral health, brushing, flossing, helping dictate what's around. And then I think what this particular paper also speaks to is the physical parameters of aging, that if you can mitigate some of these pro-inflammatory things by staying more physically active or, having stronger bones, that may also impact, let's say, the susceptibility, right, of oral bone loss that is associated with aging. So I look at it as a susceptibility piece because you have these immune changes already. Mm-hmm. At this point in time, TTP is sort of let's call it an interesting protein of interest that we can potentially target, but not presently. Yeah. But it helps us understand that with aging, we have these immune cell parameter changes that would increase bone loss in long bone and, unpublished data in oral bone. So I know that those are all sort of parts of this story, but what am I doing? I'm not eating anything different. I'm not changing too much of my diet. I'm simply trying to, balance everything because it's impossible to reverse all the aging.
Right. You just need to stay on top of the maybe inflammation. Mm-hmm.
Yes. And then from the obesity perspective, I think that, GLP-1 agonists have changed our world. They've changed our gut. We don't know how that necessarily impacts yet change in susceptibility to periodontal therapy, but those are questions that we're certainly trying to think about and ask in prospective clinical trials. But we do understand that obesity increases the susceptibility to oral bone loss. We also understand that obesity and changes with obesity increase MDSC, increases it systemically, and we have increased oral bone loss from obesity. And so trying to understand how weight loss changes susceptibility to periodontal disease is part of it, but also how it changes response to therapies, whether it be scaling or root planing and or surgery. Those are questions that I'm really interested in. So TTP is an interesting molecule, but it sets me up to understand and think about, I would say, more real-world implications of obesity and aging on this population of cells.
Fantastic. I'm loving this work. I'm excited for your next publication. And yeah, I'll follow up with Patricia. That would be fun.
Yeah, I think Patricia would be a great for the audience here. Yeah.
So you mentioned at the beginning, before we introduced you, that you were doing some fun things outside of lab. Tell us a little bit about what you're doing outside of lab that's fun.
Well, if I'm on video, everyone can see that I look like I just came out of a suntan booth or something, but I did have some therapy for precancerous lesions because of the lifelong quest to become a better tennis player. So I think that, I love tennis. My son played Division I tennis. It's part of my life. I think it's part of staying healthy and younger too.
Yes.
Back to the story, tennis players do live longer. That's actually been shown. So it just, I'm trying to stay with that. But I think, what I'd like to do with that is just sort of stay active, play a lot, potentially either teach tennis again, because I taught when I was in college, or simply become a referee for college tennis, because that's a passion of mine.
That would be incredible. I would love to see you do that.
I'm on my way.
Jonathan, do you have any other questions?
Yes. First, thank you, Keith, for joining us here on the pod and sharing a bit more context and just history with your most recent publication. And
thank you.
All the potential implications for it. Looking forward. I won't plug any more microbiome questions, but outside of tennis, are you consuming any specific media that has like your interest right now, whether it's a book, TV shows, movies, video games?
Well, I mean, my wife hates it, but I watch too much tennis. So I just watched all the French Open, and now it's the grass season. And today, in fact, Serena is coming back to play doubles in a warm-up tournament for Wimbledon, because now the grass season takes place. So I would say, Jonathan, I like to watch tennis way, way too much. So that's probably what consumes me outside of all of this.
I get it. Tennis, it's just, I feel like the last 2 Grand Slam seasons have been exciting, especially with like Alcaraz, Sinner, golf.
Yeah, and I like right, and I like to watch the next generation. So, I think that's incredibly important that for this particular sport that we have a next generation. And there's even a young man, 16-year-old kid from Buffalo, actually was a finalist in the Junior French Open boys. So go Buffalo.
Very cool.
Wow. Incredible.
Yeah.
Jonathan, didn't you go to the last Open?
I did not go to the last Open. I wish, but adjacent to that is World Cup season.
Yeah, that's true. So go USA.
Yeah.
Indeed.
Okay.
Well, thank you so much for coming on the pod today. It's
Thank you.
a delight to see you. So thank you.
I'll look better the next time.
You look fantastic. He looks like he has a little sun-kissed face. He's great.
Thank you.
Let's get horrible.